Hello there. Welcome to Primer lecture series presenting by Dr. Prem. That's me. Today we will learn about reverse transcriptase and its other enzymatic activities and how they are important for viral genome replication in host cell. So we are very much familiar with the reverse transcriptase. Yes or no? Reverse transcriptase means the enzyme which can synthesize the DNA from RNA template, which is a quite reverse to the transcription. Transcription is RNA to DNA to RNA. Here, reverse transcriptase can synthesize the DNA from RNA. But that's not the only thing it does in the host cell, but does the more than that. So that's we will learn in this lecture. And I want to ask you one question before going into the presentation that you see which one of the following enzyme activity is not found with the reverse transcriptase? DNA dependent DNA polymerase, RNA dependent DNA polymerase, RNase H, and finally RNA dependent RNA polymerase. So the question is, which one of the following enzymatic activity is not found with the reverse transcriptase? So DNA dependent DNA polymerase, RNA dependent DNA polymerase, RNase H, and RNA dependent RNA polymerase. So DNA dependent DNA polymerase means nothing but DNA polymerase only, means DNA to DNA. RNA dependent DNA polymerase means RNA to DNA. RNase H means it's an enzymatic activity which can degrade the RNA in RNA DNA hybrid. Whenever RNA and DNA forms hybrid, in that the reverse transcriptase can degrade the only RNA molecule. That activity is called as RNase H. And the last one, RNA dependent RNA polymerase, means enzyme is having a ability to synthesize the RNA to RNA. So this, we have to find out the wrong option in the four options. So if you go through the lecture completely, I'm pretty sure that you answered the question very confidently and whenever you face. Here you see Howard Temin is the Howard Temin and he is the David Baltimore. David Baltimore. So David Baltimore and Howard Temin, who discovered the reverse transcriptase independently and simultaneously. So Howard Temin from University of Wisconsin and David Baltimore from MIT. So Howard Temin discovered the reverse transcriptase activity from Rous sarcoma virus. Rous sarcoma virus. Whereas David Baltimore used the Raucher murine leukemia virus, Raucher leukemia, Raucher murine leukemia virus. From that he discovered the reverse transcriptase activity. And these two people and published their results and observation in a Nature journal in 1970. And later on, they are awarded with the Nobel Prize along with their teacher, that's Renato Delbach. This Howard Temin, David Baltimore, and Delbach is their teacher are awarded with the Nobel Prize by understanding the impact of reverse transcriptase on cancer and molecular biology. Right? And you can see this people contribution, and now also. So discovery of any gene and the conversion into the coding one, that is nothing but cDNA, is all because of the reverse transcriptase. And to understand the reverse transcriptase other activities, we need to understand the viral genome replication. That's a Rouse sarcoma virus genome replication. If you understand that, and we will be knowing why the reverse transcriptase enzyme is having more than one activity, how it is important for the viral genome replication, right? So you know this is what is the viral genome. That's a minus strand. That's a minus RNA strand. Minus RNA strand. So in this minus RNA strand, U3 and R, they are considered as long terminal repeats, And long terminal repeats, U3 and R and U5 are there at the 5' region as well as the 3' region. And there are poly A also there at the 3' region. In addition to this long terminal repeats and poly A, there is a region called as a PBS. That's a primer binding site. That's at the 5' terminal. And there is a PPT called polypurine tract. The PBS and PPT acts as a primers for first strand and second strand DNA synthesis from RNA. PBS acts as a primer for first strand DNA synthesis, and PPT acts as a second primer for second strand DNA synthesis. You see, like any other polymerase, reverse transcriptase also requires the primer, which is a complementary DNA, complementary molecule which can bind with the template wherever there is a complementary region, right? So, like any other polymerase, reverse transcriptase requires the primer. So here, viral replication takes place in the host cell. So viral tRNA, viral tRNA acts as a primer for DNA synthesis from RNA. So tRNA, which is having the complementary region to the PBS of minus strand of virus, and the tRNA binds the PBS region of the PBS region, right? So whenever tRNA binds the PBS of minus RNA, minus strand of RNA of Rouse sarcoma, then there is a DNA synthesis begins. So this DNA synthesis begins, and you see here the enzyme activity used for the synthesis of DNA from RNA is RNA dependent DNA polymerase. So reverse transcriptase is having the RNA dependent DNA polymerase. You see, and tRNA is used as a primer, and since there is a the reverse transcriptase is amplifying the from PBS to R at the 5' end region, right? Now, what will happen once it is synthesized at the 5' region, right? From here to here, right? Then next, RNaseH. You see, this is a DNA and this is a RNA. This is a RNA and this is a DNA. So RNA, this is a hybrid molecule. This is a hybrid molecule. Now, RNa's activity of reverse transcriptase will break down the RNA part in the hybrid. So it will degrade this one, right? So once it is degraded, that double strand, RNA strand in the double hybrid, then what will happen? The tRNA and amplified R and U5 fragment goes and binds to the 3' region of the 3' region of the viral RNA. So you see, this R is complementary to this one, and you have R here and here. Once it is degraded, this combination chimeric tRNA and R fragment containing one goes and binds to the R region at the 3' end, right? Then, then the first strand cDNA synthesis takes place. First strand cDNA synthesis takes place again. Here, RNA used as a template and DNA is synthesized. That is called as RNA-dependent DNA synthesis and RNA-dependent DNA polymerase activity. So as of now, we have seen the two activities. One is RNA-dependent DNA polymerase activity and RNaseH activity, right? You see, the now, so once the DNA synthesis comes up to the polypurine tract, right? Right? So, and it synthesizes the entire region up to the PPS, right? Now you see this PPT, polypurine tract in the RNA region, that's what is remains, and this RNaseH and DNA synthesis. Then this PPT acts as a primer for synthesis of second DNA strand, right? So you see, the second DNA strand synthesis takes place, and the second strand DNA synthesis means what? DNA to DNA, DNA to DNA. So you see, this is what is that DNA to DNA means DNA dependent DNA polymerase. DNA dependent DNA polymerase. In this entire replication, in the entire scheme, where three activities are required. One is RNA dependent DNA polymerase, means from RNA to DNA, and RNase H activity that's required, that degrades the RNA in the RNA DNA hybrid, and third activity is DNA dependent DNA polymerase for the second strand DNA synthesis. So with these three activities, viral genome that's in RNA in nature is converted into the double strand DNA. You see, this is what is the double strand DNA. So once the viral genome, when it enters into the host cell, it is in the RNA in nature, right? And it synthesizes the it synthesized its RNA into DNA, right? Then this double strand DNA goes and integrates into the host chromosomes, and then it initiates the carcinogenesis in the host. So finally, I'm going to tell you that viral reverse transcriptase is having three activities. That's the RNA dependent DNA polymerase, RNase H activity, and DNA dependent DNA polymerase. So you see, synthesis of viral double strand DNA from single strand RNA by viral polymerase, which is having RNA dependent DNA polymerase, RNase H activity, and DNA dependent DNA polymerase. So I hope you understood these three activities, and these three activities are very much important for the viral genome replication in the host to convert the RNA into double strand DNA. That double strand DNA go and integrate into the host chromosome. Okay. Now we will go to the next slide, where we have to prove that RNA viruses or RNA tumor viruses polymerase is having three enzyme activity, and also does it have the RNA dependent RNA polymerase activity or not, right? So now I'm actually erasing all the writing Fine. See? Yeah. So this experiment was conducted by David Baltimore and Howard Temin to prove that polymerase, which they identified in the Rau sarcoma virus or Raucher-Murine leukemia virus, they proved that they proved that this enzyme is not having the RNA dependent RNA polymerase activity. Means, so they have used the RNA polymerase assay. RNA polymerase assay. For that, what they did, so they have isolated the purified preparations of Rau sarcoma virus virions. Purified preparations of Rau sarcoma virus. So Rau sarcoma virus is a RNA virus. They have purified, they have isolated and purified the Rau sarcoma virus virions and incubated in the RNA polymerase condition, RNA polymerase assay conditions. So polymerase assay means what? So you, by using the polymerase active assay, you can detect the enzymes or proteins which are having the ability to incorporate the radio labeled nucleotides into the DNA elongation process. Right? Here they have taken the purified virions and they're treated with the non-iodate P40. It's a detergent which permeabilize the virions or virus envelope. It forms the pores in the virus envelopes, which allows the passage of nucleotides, right? So that's what. And they incubate 4-ribonucleotide triphosphates and MgCl2. So when they incubated the virions in a RNA polymerase assay condition and they incubate it for an hour or so, and later on they want to check the incorporation of radioactivity in the virions. So if they have it, if the enzyme is having the RNA dependent RNA polymerase activity, then what will happen? So one of the one of the nucleotide is a radio labeled. That's a thymidine. You know, one of the radio. So what will happen whenever RNA to RNA synthesis takes place? Radioactive ribonucleotide is incorporated. Then virions will have the radioactivity. But what is happening when they incubated in the RNA polymerase activity? They could not find a radioactivity in the virions. You know, they cannot find a. It means that reverse transcriptase, that is reverse transcriptase is not a RNA dependent RNA polymerase. It's not having the RNA dependent RNA polymerase. So with this one we can say the enzyme reverse transcriptase is having the RNA dependent DNA polymerase, RNase H activity and DNA dependent DNA polymerase, but not the RNA dependent RNA polymerase activity. You see, this is what this is what a schematic representation of Murine leukemia virus reverse transcriptase structure. So you see, this is what is a 371 amino acid and a single polypeptide. And if you look at the domain structure of the this enzyme reverse transcriptase from the Murine leukemia virus 1, and you see in the N terminal is having the polymerase activity. That polymerase also RNA dependent DNA polymerase, DNA dependent DNA polymerase. and there is a connection between and the C terminal, it is having the RNase H activity. RNase H activity. Totally, so the enzyme is having three activities. One is RNA dependent DNA polymerase, that's a reverse transcriptase, and DNA dependent DNA polymerase. It can synthesize the second strand also from the first strand of DNA. And third one is RNase H activity. That activity degrades the RNA molecule in the RNA and DNA hybrid, right? Now we can answer the question. So which one of the following activity, which one of the following enzyme activity is not found with reverse transcriptase is, reverse transcriptase is having DNA dependent or DNA dependent DNA polymerase, RNA dependent DNA polymerase, RNase H activity, but not RNA dependent RNA polymerase, right? Hope you understand the various enzymatic activities of reverse transcriptase, right? And if at all you like the uh, explanation and the video, and try to subscribe for more updates. Okay, thank you all. See you again with the one more video. Don't forget that my channel name is Dr. K. PREM-PRIMER. Right now I'm working on RDNA topics, and soon I'll be extending to the other subjects also. Thank you all, and do subscribe for regular updates. Bye-bye. Until then, next video.